You've got one molecule you like, a known inhibitor, and a big pile of others. Which ones look like it? That question, find the neighbors of something that works, is one of the most used moves in real drug discovery. Today we answer it in code, using the fingerprints and Tanimoto similarity we built back in episode 2, on the drug-like set we filtered in episode 4. The recipe is simple. Take a query molecule, here, gefitinib, a real approved EGFR drug. Turn it into a fingerprint, turn every molecule in your library into a fingerprint, then score the query against all of them with Tanimoto. Sort by that score, and the top of the list is the molecules most like your query. Query, fingerprints, Tanimoto, top hits. First the query. This is gefitinib. You can see its structure on the right, that fused quinazoline core with a chlorofluorophenyl group hanging off it. We fingerprint it with the same Morgan generator from episode 2, then fingerprint all 800 plus molecules in our drug-like set. Everything is now a barcode we can compare. Now the search. BulkTanimotoSimilarity scores the query against every fingerprint in a single call, fast, even for thousands. We drop exact duplicates, sort descending, and take the top 6. Look at the scores, 0.92 at the top, dropping through the 80s. Those are strong matches, molecules that share most of gefitinib's chemistry. And here's the payoff, the top 6 neighbors, drawn out. Look what similarity search found, every single one carries gefitinib's anilinoquinazoline core, the same chlorofluorophenyl, the same fused ring system, differing mostly in the solubilizing tail. And they're potent, pick 50s of 7 and 8. That's the whole chemical series, surfaced automatically from a barcode comparison. One more view, the similarity of the entire set to gefitinib. Almost everything sits on the left, unrelated molecules with low scores. Only a small tail climbs above 0.7, the analogues. That shape is typical, most of any library is noise, and similarity search is what pulls the signal, the true neighbors, out of it. So with fingerprints and one Tanimoto call, we went from a single known drug to its whole family of lookalikes, the engine behind lead hopping and analog finding. But so far we've only searched. Next episode, we stop eyeballing and start predicting, training a machine learning model that guesses potency straight from a fingerprint. See you there.